everyone, and welcome to today's Lab Tools webinar. I'm Nikki Spahich, Scientific Technical Editor for The Scientist, and I'll be moderating our discussion. Today, our speakers Christine von Raesfeld and Dr. Stephanie Cullen will be discussing research experiences from researcher and patient perspectives. And with that, let me introduce our first speaker, Christine von Raesfeld. Christine is a leader in bringing a critically needed patient perspective to cutting-edge medical innovation. A patient advocate committed to providing patients with chronic and rare diseases with the support they need, Christine works with patient advocacy organizations, industry representatives, and individual patients and their families. Christine?
Thank you for that introduction. Gosh, it makes me sound really busy. So I wanted to say thank you for including me, and thank you to Sanguine for giving me this opportunity to share my perspective. So what we are talking about today is simplifying the patient participation in research. And before we begin, I'm going to tell you a little bit about me and a little bit about what I believe. So before I start, I just want to go over the difference between empathy versus sympathy. And I'll let you know why in just a second. But empathy is feeling with or alongside someone, while sympathy is feeling sorry for them. So the reason I'm telling you that is because my story is a long one. It is dramatic and scary and everything else that you wouldn't want to happen in your life. But with that story, I don't want you to feel sorry for me. I want you to feel empathy and to feel with me. But don't feel sorry for me because I have an amazing life and I've done some amazing things because of everything that has happened. So, who am I? Like everyone else, I am a million things to a million different people. So I'm a daughter, a sister, an aunt. But the main thing is I'm a person with my own beliefs. I have my own feelings and my own voice. And I hope that by using my voice, I can help to spark the change that we need in healthcare today. So medically, me, I have been diagnosed with multiple conditions. I have rare disease. I also deal with multiple autoimmune issues, including CNS lupus. So I had periods of time where I went through psychosis and I had a psychotic break. I was hallucinating, but managed to work my way back to some sort of normalcy. So I advocate for chronic illness, rare disease, mental health, anything that has affected me in some way in healthcare, I am willing to speak out about it. And I use my experiences to try to evoke the change that we need here in healthcare. So my healthcare journey, it's a long one, it's elaborate. You'll see the pictures on the side of the screen, which kind of show you where I'm at. In the top left corner is the brain damage I have from toxic encephalopathy, which is poisoning from medications. I've had 3 joint replacements, both my knees, and a myriad of other issues. So my chronic illness journey began at a very young age. I was 5 years old. And since then, I've had multiple diagnoses, several misdiagnoses, and long diagnostic time. My case management was not what it should have been. I have no real history of my disease. I was with a hospital that when they switched over to electric medical records, just happened to lose all of mine. So we recently found 5,000 pages, and I'm working my way back into finding out why I've stopped medications and basically my whole health care journey. I have diagnoses in there that I didn't even know I had until we found those random papers. So I also deal with mental health issues, the medication toxicity like I explained before. I do have toxic encephalopathy, 3 joint replacements, and I've lost night vision, all due to medications used to treat the symptoms of my disease, not any of my diseases specifically. I've had negative experiences with industry and nonprofits, which has led to some mistrust and general negative feelings sometimes in this space. But it has led me to some good things. I am part of some precision medicine initiatives, and I am a patient in Stanford's Humanwide program. I'm sorry, I'm getting over a cold, where I have done my pharmacogenomics, where we may have lost my records, but I do know now that there are certain medications I can't take because of that test. All of my experiences also brought me into this national advocacy where I've been speaking up on patients who, like me, are disabled, on Medicare, and need options. So my own experience with research and trials started when I was young. When I was 14 years old, I was diagnosed with a rare blood disorder, thrombotic thrombocytopenic purpura. And at that time, I did everything I could to find out about my disease. I would take the bus over to Stanford and I would sit in the medical library for hours. I had this desire to get better and to improve my life. When things started happening and my diseases started to go downhill and there was no options for some of the things that were affecting me, I decided that it was my desire to help everybody after me, including my nieces, who one of them may be showing symptoms of an autoimmune. So at the time, and ever since my diagnosis, it's been difficult. Clinical trials, I've always wanted to participate, but if you've ever been to clinicaltrials.gov, you know it's not an easy place. So it was hard to find trials that had qualified. I had no real resources or knowledge. I was with a hospital that didn't even offer clinical trials, so I knew basically nothing about it. The only resource I had was Stanford, who had come in because I had a rare disease, and they were looking at me from that perspective. So the way I found help, I started connecting with my community and different organizations like Sanguine. I actually met Sanguine at a walk a few years ago, and from then started a relationship with them. They had taken my information and my conditions, and anytime they had a research opportunity or a trial that pertained to me, I would get a call from their staff. I've been in 5 trials because of Sanguine, and a few more with my own hospital now that I've learned to advocate for myself. So considering the patient perspective, if we're looking at simplifying the whole clinical trials process, the first thing that I've noticed and what we've talked about with several people is awareness. So most patients aren't even aware that clinical trials are an option, and they don't know what they actually are. People think that clinical trials are somebody mixing up medications on a side room and there's no testing and that they can die. People think clinical trials are only for people who have no other options. Second, another issue that we have is access. So it's difficult for a patient to find trials, let alone go through the whole enrollment process. If you've ever seen clinicaltrials.gov, you know there's a series of inclusions and exclusions, and most patients don't know the difference between any of that. And there is also the issue of trust and fear. So there's a general mistrust in the patient community when it comes to pharmaceutical companies and to healthcare industry. As you know, a lot of people don't tell their doctors everything. There was just a hashtag recently going around of patients are not faking. So there's a lot of issues there that really need to be worked on. Also, cost of clinical trials. If an insurance company isn't covering the clinical trials, most of the patients who use that as an option aren't able to afford that as well. And there's also the inconvenience of trials. So not only the time that it takes, the transportation, the gas. I personally am on Social Security disability, and my budget is limited, and paying for things like gas to get to a trial isn't going to be one of my priorities. So, moving on, the solution that I think is that we need to bring back empathy. So to ask first, patients want to be involved, and many of them are willing to participate, but they've never been asked, and they don't know their options. So we need to educate patients. They're more willing to participate if they're informed and they're aware, and they know what's going on. Engage them, become a part of the community, and give patients a reason to participate. One of the main reasons why I got involved was to give myself a purpose. I had spent a year in bed hallucinating and just kind of living this weird, strange, alternate reality. And the purpose and giving people something to hope for is what got me out of bed. So meet patients where they are, make the process simple. If you minimize the burden on the patients, you're going to maximize your outcomes. And by empowering the individual patient, you're empowering an entire community. Other benefits of simplifying this for the patient is better patient engagement, and it's cost effective. Most patients get most of their information from other patients. I myself know that I receive emails from nonprofits and organizations that I work for, and I don't really look at all of them. But I do know if I get a message from another patient who's done something, I'm more willing to look into that. You're also looking at improved data capture and better research. And so last, concierge services like these offered by companies like Sanquin's, I think can help increase the numbers of participants in clinical trials. By making the process easy and transparent, you lessen the burden for the patients, and ultimately you build trust. And that's what we need here. You also ensure adherence when someone else becomes accountable, and you build a community committing to finding cures. So whenever it comes to working with patients and getting involved with patients, make it easy and make it worthwhile. Make sure that your patients know that there is a purpose and there's a reason. And that's all I have for you today. So thank you for giving me the time to speak. And if anyone has any questions after we're done, I'm happy to answer.
Thank you, Christine. As a reminder to our audience, you may ask questions using the question box at any time. Our panel will have a chance to address these after our next speaker. And that next speaker is Dr. Stephanie Cullen. Stephanie is the co-founder and CEO of Persephone Biome, a business taking a novel approach to improving the efficacy of current cancer therapies, leveraging the systemic impact of the gut microbiome on the human immune system. Persephone's technology platform is based on collecting and analyzing gut microbiota samples from thousands of cancer patients, then using machine learning in conjunction with systems biology tools to understand the impact of gut microbes on patient prognosis. The results are used to design novel immunotherapies and companion diagnostics for cancers of unmet needs. Dr. Culler is a recent graduate of the prestigious Y Combinator Accelerator and has spoken at TEDx San Diego. She received her PhD in chemical engineering from the California Institute of Technology under the mentorship of Professor Christina Smolke. Dr. Culler has had over a dozen publications and patents on synthetic biology and microbial-based technologies. Stephanie?
Thank you very much. And I'd like to thank the scientists as well as Sanguine Biosciences for the opportunity to share our story with you today. I'll be getting into some data shortly. But Christine, thank you so much for your inspiring story. Like you, while haven't been a patient, the inspiration for starting Persephone Biome was really from the loss of both of my grandmothers to cancer over 2 decades ago, and inspired me to become a scientist to eventually start this company and form the mission around enabling a lifetime free of cancer. So at Persephone Biome, we're developing a technology platform that we call Decode, Design, Cure. And this brings together artificial intelligence with systems biology tools to rapidly develop what we call microbial-based immunotherapies to enable our vision. We are developing essentially a cocktail of bacteria that are therapeutics that can be taken in combination with cancer therapeutics. The following slide is essentially what we're focused on. So in the field of cancer, there's been really a breakthrough with the discovery of immunotherapy drugs, specifically checkpoint inhibitors. And as many of you know, these are a type of immunotherapy that essentially awaken the immune system and allow it to recognize, attack, and kill cancer quite effectively. They have been approved to treat over 40% of cancers, which is a very significant patient population in the United States alone. And as we know, they can result in complete cures. We have cures for cancer. The main problem is that 70% of cancers 70% of cancer patients fail to respond to these therapies. And for patients, similar to what was just discussed earlier, these therapies are very expensive, over $150,000 per year. And failures all not only lead to lost lives, but for pharma in particular, this is a lot of money that's being left on the table and lost in potential revenue. And in addition, what's been found through back-to-back science papers early last year, that cancer patients who fail therapies, the 70 to 80% of cancer patients are missing crucial bacteria in their microbiome. We at Persephone Biome essentially have two solutions. One is to put back those missing microbes. So we're developing what we call microbial-based immunotherapies that fully enable the curative power of checkpoint inhibitors. Second, in order to really maximize the efficacy of our therapeutics, we're developing what we call microbial diagnostics, essentially a simple, non-invasive diagnostic based on a stool sample from these patients to enhance the efficacy for everyone. So the gut microbiome, just as a quick refresher for all of you, the microbiome is a collection of all the microbes that live within our GI tract. And what's important here to recognize is that truly a healthy gut microbiome prevents and fights cancer. That's the role of the microbiome, including many other diseases. But now we've come, through thousands of papers and publications in the literature, we now know that damaged microbiomes, or what we call dysbiotic microbiomes, are associated with disease, including cancer, diabetes, and Parkinson's. And every day there seems to be another connection to a notable disease. On this slide, we have a schematic here that represents the key findings in the field of cancer immunotherapy and microbiome research. So I mentioned that there were 3 seminal science papers that came out last year. These papers together demonstrated that gut microbes very much impact patient response to checkpoint inhibitors. What was found and shown on the left-hand side is that for the majority of cancer patients, those that are non-responders, so 70 to 80% of cancer patients who fail therapy, they are missing crucial gut microbes. And as a consequence, they have a severely impaired immune system, and it doesn't allow them to respond to these checkpoint inhibitors. This is in contrast to the 20 to 30% of cancer patients that do respond to checkpoint inhibitors. They essentially have a fully functional microbiome, quite diverse. So lots of different kinds of gut microbes, diverse in function, and their immune system is on. It can respond to the checkpoint inhibitor. Our goal at Persephone Biome is to convert these immunotherapy non-responders into becoming responders with what we call microbiome medicines, or as the FDA has termed our product, a live biotherapeutic, which restores these missing gut microbes to turn on the immune system. We want to go from 20 to 30% potential cures to 50% or higher. That's the mission at Persephone Biome. And in order to do that, we've built an artificial intelligence-driven drug discovery platform that very much leverages patient data, patient specimens, that we collect through efforts with Sanguine Biosciences as well as other partners. But patient specimens is really what drives our research. Without it, we wouldn't be able to develop the therapeutics that we're looking to. This is really, first step in precision medicine for our field, and, being able to access a wide variety of patients for our research is really critical. And so how we use artificial intelligence, we namely use it right now to discover the missing gut microbes in these cancer immunotherapy non-responders. So at a very high level, to walk you through the schematic in our technology platform, that really allows us to go rapidly from patient data to first-generation therapeutics, and hopefully in the clinic in the next year and a half, we begin the journey with patients. We begin the journey with their specimens, namely stool, which is a reflection of what's happening in the gut microbiome, as well as blood samples to understand what's happening in their immune system and how they're responding to immunotherapy. We also collect the same samples longitudinally from healthy individuals so that we can compare the results from cancer patients to those that do not have cancer. We then perform something that's called decoding, and this is where systems biology tools come in, namely omics techniques like genomics, DNA sequencing, transcriptomics, RNA sequencing, proteomics, essentially protein sequencing, and metabolomics, looking at all the metabolites. We perform these analyses on both the stool and the blood. And what that allows us to do is essentially unlock what we call the metabolic relationship between the microbiome, the human microbiome, and the immune system. We identify the bacteria, their function, the metabolites, and how that impacts immune response. All of these complex data sets, or omics data sets, are then fed into machine learning. The machine learning then rapidly looks for essentially patterns that are unique in these cancer patients that are non-responders. With this, we're able to then discover what are the missing gut microbes and their function, or how does that impact in patients' response to immunotherapy. We then further use computational models to define and essentially refine the therapeutic, and then go into the lab and build our consortia. These are rational mixtures of gut bacteria that have been isolated from healthy individuals. Through these efforts, we've collected and discovered over 1,000 strains of gut bacteria that we can use as the inputs to our therapeutics. And we iterate this approach, going through and validating our therapeutics both in vitro and in vivo in mouse models of cancer until we have a very robust therapeutic. Now, on the following slide is an overview of our campaign called Poop for the Cure. This is a nationwide collection of whole stool samples for our research. And the reason why we initiated this campaign was because of the difficulty in accessing stool samples. There are no biobanks for stool samples that exist today. There are no standardized techniques. This is not something that's implemented as a standard sample that's received from patients on clinical trials. There just does not exist an infrastructure. And as we started this effort of collecting stool samples from cancer patients as well as healthy individuals, we quickly realized that the kit we were previously using was not sufficient. It was difficult to use. We had a lot of complaints. And really thinking with the patient in mind, we wanted to take the stigma out of providing a stool sample. We wanted to make it easy to use, and we wanted to make it fun. And also educational. We want people to understand that there's a tremendous wealth of information in a stool sample, and we're just beginning to unlock what that data means. But in order to do so, to potentially develop therapeutics and diagnostics and other potentially down the road consumer applications, we need to be able to get these samples. And so that's how we came up with Poop for the Cure, and redesigned our kit, as you can see, to look like a luxury handbag. And we have online a questionnaire that examines a person's medical history and allows us to bring in thousands of stool samples to really enable our machine learning platform. We are looking with Poop for the Cure for stool samples from anybody with cancer, those patients that are in remission from cancer, as well as healthy individuals. In return for a stool sample, we provide the donor $50, a $50 gift card, as well as a very cool Poop for the Cure t-shirt. And we implement this kit on our observational clinical trials, which I'll describe shortly. So on this page is a dashboard of the efforts that we've conducted to date. Again, we need thousands of patient samples in order to really perform and develop precision medicine. And as you can see, we've had very good success with Poop for the Cure to date. Over 1,500 patients enrolled. It's growing daily. We have coverage from the entire United States, which is really critical when we're talking about designing therapeutics for diverse patient populations and making sure that our therapeutic will work for both men and women and individuals with different ethnicities. We have collaborators, as I mentioned. Sanguine Bioscience has been very critical in really enabling our science, going from an idea in a project proposal to getting patient samples within a very short amount of time. Other efforts take months to potentially more than a year to get the infrastructure in place to collect samples from patients on clinical trials as well as at home. And so very thankful for that relationship. We also have ongoing observational studies with UC San Diego Moores Cancer Center as well as City of Hope. These studies are slightly different than Proof of the Cure in the sense that they're longitudinal. So these are studies that go on for months, if not years, where we collect every few months a stool and a blood sample from a patient. And that's really critical when we go to talk about designing our therapeutics and what's needed there. Well, also on this dashboard, I have a little bit of a sample of the types of metadata. We have samples from a diverse set of cancer types, so pretty much all the top cancers as well as those that are rare, and a good subset of patients who've been receiving the FDA-approved checkpoint inhibitors. This is all critical because as we go to design therapeutics for a number of different types of cancers, we have a large database which we can mine to start the development. And then finally, going back to patient advocacy, the patients are the ones who really support our research. Much of the growth in terms of donations has come from our patient support groups nationwide. We can't thank them enough for their help. And then this is an overview of just the types of longitudinal studies. I would say 70% of our efforts at Persephone Biome are from collections that we've received through longitudinal manners. And longitudinal collection is very, very critical because it allows us to understand the changes in the immune system as well as the microbiome of these patients over time. And that helps us better understand two things. One, designing our therapeutic, we want to understand the needs of the patient throughout the course of treatments as well as their cancer. And two, to develop robust therapeutics. We want to be able to take a stool sample before they receive immunotherapy to better predict if they will respond to that drug, but also how can we find the right patients that will be most likely to respond to our therapeutic. That's really the goal here, to maximize efficacy. So getting into the technology, I want to describe a little bit more about our decoding because I'll present some data shortly. So as I mentioned earlier, the real focus here is to decode the difference between non-responding cancer patients on the left and those that are responders on the right. We really want to understand the metabolic relationship between the microbiome and the immune system that enables response. So what are the microbes, what do they produce, what are the metabolites they produce, and how does that impact the immune system, and in this case, cancer patients' response to immunotherapy. So essentially on the right-hand side, if you look at the schematic, we are trying to use these techniques to map out the mechanism. We're mapping out through genomics or deep whole genome sequencing. We're validating and discovering what are the microbes in the stool samples of these patients. Through metabolomics, we find the metabolites. Through immune profiling or immunophenotyping, we then understand the immune response. And then through either patient-provided data or physician-provided data, we understand their response to the immunotherapy drug. So the very first step in our process on trying to discover what's truly critical in the microbiomes of these cancer patients is to perform whole genome sequencing, essentially DNA sequencing, on the stool samples that we collect. So from these patients, essentially the stool sample has a collection of gut microbes that reflects the environment that exists within their colon. We then provide DNA sequencing, perform DNA sequencing, and from that, we then have a DNA sequence that we can mine to see what's different between cancer patients that are non-responders than those that are responders. And from that, we've been able to, through analysis of several hundred patient samples, identify about a half a dozen to 16 or so gut bacteria that are either clearly missing or very low abundance in these cancer patients. We believe that these are the microbes that are responsible for patient response, and these are the microbes that essentially make up our therapeutics. On the following slide is results from whole genome sequencing of stool samples from 3 particular patients. Each pie chart that is shown here is the result of whole genome sequencing, so DNA sequencing of these stool samples. The pie represents the entire microbiome composition from somebody, as well as each slice of the pie represents a different gut microbe. And what you can see with the cancer patient who's a non-responder on the left-hand side, few things. One, they have very low in diversity, so limited kinds of bacteria. But what's been striking that we've observed consistently throughout our data set with cancer patients who are non-responders is that one or two microbial species essentially dominate their microbiome. And this is concerning because these species can be highly inflammatory in nature. Some cause cancer, those that have been actually connected to carcinogenesis. And finally, many of them are pathogens. And we see this pretty consistent with the advanced cancer patients who are non-responders. And what's striking to us is that if this information today is not provided to patients. There, again, as I mentioned, there's no infrastructure for being able to review this type of data with a physician and have an actionable insight. That's really the goal of our efforts at Persephone Biome, is not just to develop therapeutics or diagnostics, but to have some type of actionable insight from this type of data. Next to the patient who's a non-responder, you have a microbiome sample from a cancer patient who has responded very well to immunotherapy. In fact, they are currently in remission. You can see it's very diverse microbiome, I would say very similar in nature to that of a healthy person. As you go to the next slide, what we have discovered, as I mentioned earlier, we have discovered that these cancer patients are missing crucial gut microbes. And what I have shown here is just a display of essentially the abundance of these microbes in all of the stool samples, or a portion of the stool samples we've collected and analyzed to date using whole genome sequencing. So on the y-axis, you have microbes A through J. These are all microbes that we have identified to be statistically different between cancer patients, which are dots in dark blue, and healthy controls, which are dots in light blue. Again, each of these dots represents a whole microbiome sample from that patient, and then the amount of that microbe is shown essentially on the x-axis. We consider anything that has an abundance less than 10 to the minus 4 to be essentially undetectable. And so you can see here for the majority of these microbes, the cancer patients seem to have either low abundance or essentially missing these microbes. Again, these are the microbes that we've identified that we believe are the ones that we need to push forward for therapeutic purposes. And cancer patients in general are not just missing 1 or 2 microbes. It's a set of about 12 to 16. So all of the microbes I'm showing here are essentially missing, or those that are really, we think, important for immune response to checkpoint inhibitor therapy. And on this slide is a different pictorial view of the data I just showed you, but it looks over our entire data set that we've collected today. Again, this is from whole genome sequencing, and this is what we call a volcano plot. On the right-hand side are bacteria that we have found to be enriched in cancer patients. So not only does our technology platform identify the bacteria that are probably good for you as well as important for response to immunotherapy, we can quickly identify the bad guys, so the pathogens. And what I've shown here on the right-hand side are and highlighted a few types of bacteria that have been known to either cause disease or are highly inflammatory. I have highlighted Clostridium difficile, a very infectious microorganism, as well as D. fragilis, which produces an endotoxin that causes inflammation. On the left-hand side is bacteria that are enriched in healthy individuals. And what I've highlighted are some of the missing microbes. And so the y-axis here shows statistical significance, and what I have put into the boxes are essentially those that we find to be very important between healthy individuals and cancer patients. So then how do we use artificial intelligence to then look at our data and drive the development of our therapeutics? Essentially, we use artificial intelligence to predict our therapeutic compositions. So as I mentioned earlier, we've discovered missing gut microbes that we think are very important for patient response to immunotherapy. Well, there's quite a few of these bacteria, and that's a pretty large sequence space. So we use essentially artificial intelligence to look at combinations of gut bacteria that when added in silico, so computationally, added back to a stool sample that is a non-responder, what are the combinations of gut bacteria that can convert that non-responding stool sample into becoming a responder, right? So what are the therapeutic combinations of gut bacteria that can convert somebody from becoming a non-responder to a responder? And essentially, that's what the AI algorithm puts out to us. The output of the algorithm is a set of therapeutic candidates that have mixtures of gut bacteria that it shows computationally can do that conversion. So from us to reduce how many, the number of combinations that come out from artificial intelligence and reduce it to a size that we can, experimentally validate both in vitro and in vivo in mouse models of cancer, we look at the gut bacteria that are selected most often by the machine learning. And that then becomes essentially our top gut microbes that we then validate as our therapeutics. We typically validate therapeutic compositions on the order of 4 to 6 gut microbes. We don't like to go higher than that simply from a manufacturing standpoint. It becomes technically very challenging to do that and can cause issues when you're trying to scale for clinical trials. So in the following slide, we wanted to show a few things. One, that there's a causal relationship between the microbiome and response to checkpoint inhibitors. We also wanted to show efficacy of our therapeutics in converting a non-responding microbiome into becoming a responder. And so in order to show causation, we performed a fecal transplantation from a cancer patient who's a non-responder to immunotherapy and transplanted that into a cancer mouse model. We, in conjunction, administered a checkpoint inhibitor, and we looked for the response in the mice. And as you can see on the left-hand side in red, the mice essentially who were fed this fecal transplant from a non-responder became a non-responder. They had a very low response to the checkpoint inhibitor. As you can see, large tumor volume signals low response to the checkpoint inhibitor. And then we wanted to see if co-administration of our therapeutic, PB327, a consortia of 6 gut microbes fed orally to the mice, those that were fed the non-responding fecal material, could we rescue the response? Could we convert this human non-responding microbiome into becoming a responder? And indeed, you could see the case. And so this was a very important study for us because it proved the translational proof of concept that we'd like to achieve in the clinic, that we can treat a human non-responding microbiome into becoming a responder. And this is actually how we validate our therapeutics. Again, being very patient-focused. The design of our therapeutic is based on patient data, real-world patient data. How we validate our therapeutic is in the background of a human non-responding fecal sample. And that's what we think is important for de-risking and improving how we translate our therapeutics to human patients. So in summary, our technology platform, the Decoding, has allowed us to rapidly discover the key missing microbes from cancer patients who are non-responders. We've been able to use our design technology to create a therapeutic that contains those missing microbes required for response. And finally, in terms of the cure aspect, we have been able to show, again, this is in mice, we've been able to convert a human non-responding microbiome into becoming a responder. And we look forward to hopefully being in the clinic in mid-2021, so in about a year and a half. We're focused on optimizing our therapeutic and achieving our IND sometime next year. So thank you for your time. Just in closing, the vision of our company right now is to treat, eventually cure, and we want to see diseases prevented. Cancer, of course, is our first focus, but we believe that we're developing a technology platform that could be applied to virtually any disease, really. Again, it's all based on patient samples, medical history of these patients to really inform the design of our therapy. So in closing, the patients are waiting, and we are focused on trying to deliver therapies in a meaningful and a timely manner for them. Thank you.
Thank you, Stephanie. The audience has submitted several questions, so let's get to them. Christine, can you briefly walk us through working with a concierge service like Sanguine? How do patients sign up and what happens next?
So I met Sanguine actually out at a community event. I was doing a walk for a local lupus foundation, and they had a booth. And the women that they had working the booth were very friendly. They knew the disease, and they were very easy to talk to. So they were offering an incentive, a cash or gift card incentive, I think it was, if you signed up and you qualified for any type of lupus study. So, of course, I was looking into that clinical trials, and they came in right at the right time. And so, of course, I signed up because anything I can do as a patient to help with research, I'm going to do. So I signed up. They contacted me the first time, and I went through with everything. What they did do, which was amazing to me, was I didn't have to do anything. They called me on the phone. They had somebody walk me through the process. They told me what they were doing. They said who it was or what they were looking to do. They asked my permission. They told me what to expect along the way. And then openly asked for any questions, completely guided me through the process. Instead of me having to go get my medical records and try to sort out everything myself, they sent me a DocuSign document. They had me sign it, and they got all of my medical records for me. On most of the ones that I've done for them, they've sent a phlebotomist out to my home for the first time and collected samples from my home, which was amazing to me, especially at times where I wasn't driving or didn't have reliable transportation. So that helped a lot there. The girls that had called me and that I was in contact with knew everything about these studies. I could call at any time with any questions, and I knew exactly what to expect. Every time they have something that I qualify for, they contact me, which is nice because I don't have to go out looking for myself. So I have done 5. I was on my 5th one back when I spoke with Sanguine last year, and I'm just waiting for other opportunities. I'm happy to give any samples. And the fact that they make it easy and come out to my home and they schedule around me, it just makes it so much simpler, and I don't have a headache of worrying about anything. So, and they're very gracious. They call, they make sure everything went well after the whole procedure and make sure that I'm happy. So, and they've been in our community. So it's nice to see a company that actually gets into the community. And when I hear about something, I tell my friends. And like I said before, I can get a million emails from a million different organizations. But if a friend or a patient tells me that they did this and they like this and these are the reasons why, I'm going to go with that.
Great, thank you. Stephanie, do we know what causes the microbiome dysbiosis in cancer patients? Is it the cancer itself, or is there a predisposition that leads to the cancer?
That's a very good question, and a question that I think a lot of individuals and researchers are contemplating and looking into right now. For us, we don't look too much into what's causing this dysbiosis. We're focused on treating advanced stage cancer patients. Most of the patients that we treat have failed therapy, prior therapy, namely immunotherapy. Some have failed chemotherapy and other types of therapies that are applied. But what we see, and there's a lot of data in the literature, is that we're seeing damage that is caused by years of various therapeutics, antibiotic use, aspects of diet and lifestyle. All of these are different factors that play a role into causing dysbiosis. It's difficult to parse out the impact of cancer or vice versa just because of the complexity of cancer, and everybody's cancer is different. We can somewhat parse that out in animal models, but still it's very, very difficult. So I would say that, really the focus has been trying to understand more so, not necessarily how the microbiome causes or this dysbiosis or vice versa. It's really how to understand what's happening with dysbiosis, how to prevent dysbiosis. I think those are more of the factors that I think researchers are focused on today.
Great, thanks. Christine, do you think it's more important to educate doctors on clinical trials or to go directly to the patients for education?
I actually think that doctors should know about them. I don't think it should be a doctor's duty. I think that it should be patients telling other patients. I personally, with my doctors, they don't have enough time. I have enough issues, and when I go in, it's to discuss my medical issues, to discuss my symptoms. I know they have limited time. They have limited staff. I'd like them to focus strictly on my health when they're working on that. So as far as clinical trials and things like that, most of that I've heard from other patients. And I get them now. I'm at a teaching hospital, so it's a lot easier. And I have my doctors over at Stanford throw me in whatever I qualify for with them. But most people don't have that. I don't know if you'd say that's a luxury, but for me it is. Most people don't have that access. Yeah, and I think the patients are the ones that need to know. They're the ones making the decisions, and they're going to listen more to patients. I think that the conversation in the patient community, I get more out of the patient community than I do in my doctor's office. A lot of times I come to my doctor's office with things that he can confirm because of things that I've said. So it's an interesting mix, but I do think the doctors have other things on their plates. I think it's nice for them to know that there are clinical trials running for certain things, but I don't think it's their responsibility. So I think patients telling other patients, it's going to get the word out there, and it's a reliable source when you're going out through other patients who have been through it and who, they're the ones that are giving their blood, sweat, and tears for these studies. And I think it's just that patient-to-patient community that we need to build.
Thanks. Stephanie, is the response to your therapeutic short-term or long-term in the life?
Well, right now, a therapeutic has always been validated in the context of preclinical mouse studies, and for the most part, the response is we only validate the response during the cancer treatment, and our focus is to having complete regressions in the mouse models, which we hope will translate to regressions in cancer patients. Our therapeutic will be taken daily, similar to how we take probiotics, for example, or like a vitamin. We believe that the activity of our therapeutics, namely the bacteria, are temporal. These are bacteria essentially that do not engraft. And so from a permanent standpoint, we don't believe that our therapeutic will have essentially a permanent effect. We're really trying to focus on enhancing the efficacy of the therapy that we're combined with. Now, that being said, this opens up the door to having a different therapy post, a different version of our therapy post that of the combination. So once somebody is in remission, we may want to develop a different type of therapeutic that can be taken chronically to really create a homeostasis. So this would likely have a lot more different diversity in the bacteria that's there, as well as those that engraft. And one other reason why we don't necessarily, for this therapeutic purpose, want our bacteria to engraft, this is a point of discussion in the microbiome space today, is that we want our modality to be very similar to traditional drug therapeutics that don't have a permanent effect on the patients. We want them to essentially wash through somebody's system in case we wanted to provide a different type of microbial therapeutic. That's really the focus and how our therapeutic works today.
And as a follow-up to that, have you looked at all at the mouse gut microbiome after the therapy has kind of gone through? Are there differences before and after that it may be lasting?
Yes, and that's what I mentioned. So we analyzed stool samples collected from mice throughout the study, as well as once dosing stops for over a month, for example, to look at the engraftment of our bacteria. And I would say a small fraction of the bacteria that we have included in our compositions engraft. And those are the ones that are essentially associated with the gut mucosa that physiology, in terms of physiology, have been associated to engraft and be a part and kind of stick to the GI tract. Those are the ones that engraft. But I would say by and large, our bacteria are transient in nature, and they wash out of the system within hours of the dose.
Thank you. Christine, based on your own experience, could you please explain further the importance of showing empathy as a patient advocate in the health field?
Do you mean showing empathy as me being a patient advocate or as somebody in industry looking at a patient advocate?
I believe someone looking at the patient.
At the patient. So I have a really quick story about that. I had gone to a rare disease drug development seminar, and I sat in a room with a bunch of executives, and they were talking about getting together and collaborating and getting a rare drug to market. And in the room, the woman had asked that was speaking had said, how many in this room would be willing to collaborate and work together? And while we sat there, nobody raised their hand. And so after the session closes, as you all know at conferences, they open it up to questions. And at the end of the session, I stood up and I said, I'm not one of you, and I'm a patient. And to sit in the room and to know that you are one of my only hopes in getting a cure for these diseases, and to know that you're going to go home to your family and your children and plan your vacation, and I'm going to go home and figure out what I need to eat or what I'm giving up to pay for meds. It really is disappointing to know that that would happen. And so I think people don't think of patients and what they're working for as patients. They don't look at us as people until illness hits somebody in their family. And then I think it puts a different spin or a different perspective. So I think you have to realize that people that are next to you and they look normal, they're also people that could be dealing with illness. And so I think that's a big, huge part that people lose focus, and it becomes about other things, and it's not about the patient. So I think just having that empathy and knowing that this person is giving everything to you. They're literally giving you their blood and their sweat and their tears and their poop, and just to look at them as somebody makes a difference. And so I think everybody in the world does things for a reason or to get something out of it for themselves. And so just look at it as, I guess they're people, and that we're not numbers and we're not samples, and that it puts a different perspective and a different spin on everything. And I think that empathy could really make a difference in building communities and even just engaging people in clinical trials, just building that trust and building these communities where you get in there and you're helping these people. And if you help and get in these communities, they will do everything to help you. So I think that just needs to be brought back.
And as maybe a little bit of a follow-up, how can really any patient better advocate for themselves in the doctor's office and beyond?
I think just being prepared, knowing what you're going into, a lot of times taking someone with you. I like to email my doctors beforehand with my concerns so that they know ahead of time, because you do have limited time with your doctors. I think just focusing on telling your story and knowing that whatever you are feeling is right and that nobody else can tell you how you feel. For a long time, when I was having the brain issues, I was going in and my blood test was fine. And I kept saying I felt off and I didn't feel right. And the doctor said, everything looks great. You look amazing. Go see a psychiatrist. And so he did. And for 2 years, I saw a psychiatrist who said, it's not my issue and you need to get checked out again. And by the time I did, I had ended up with toxic encephalopathy. So just, I think you know your body best, you know what you need, and to focus on what you need. And if somebody doesn't hear you, to go to someone else. So I think that's just the most important thing. I know a lot of people who didn't advocate for themselves and who aren't here anymore. So just to keep in mind that nobody else knows you and you're not crazy for what you're feeling.
Thank you. Stephanie, could you supplement the missing metabolites rather than the microbes as therapy?
That's a really good question. So the number of metabolites and metabolic pathways that we discovered that we believe are part of this is immense. So from a FDA perspective and a small molecule approach, it would be very, very challenging to do so. So the response and the role of the microbe seems to be pleiotropic. So multiple mechanisms that are inducing an immune response that's beneficial to these cancer patients. So that is something that we looked into initially, but the results are just so complex that it's almost infeasible that it's a single metabolite or just a few. It seems like there's many metabolites that are involved at different levels and would become very challenging from a drug perspective to pursue that route.
Okay. And Stephanie, this next question is for you. Do gut microbes impact treatments to other diseases other than cancer in the same manner by interacting with the immune system? Does anyone know anything about that?
Yes, in fact, there's been many reviews as well as some clear studies looking at that. One of the reasons I'm a chemical engineer and fascinated by I call it catalytic capacity of the microbiome. Most people don't know that there are about 100 to 300-fold more genes in the microbiome than there are in our own cells, right? So immense opportunity in terms of chemistry, and this chemistry actually impacts how many drugs work. Some cases favorably, some cases, inactivating the drugs, and in other cases, that chemistry is what's involved in how the immune system response to that therapy happens. And so what we've seen in the literature is that there are at least 50 or so FDA-approved drugs that are impacted by the microbiome through those mechanisms, some catalytic and some in ways that the immune system counteracts their effects in ways we don't know yet. But we know that this is the case for not just cancer therapies, including checkpoint inhibitors or chemotherapy, but this has happened with therapeutics that are used for Alzheimer's. This has happened with therapeutics that are used for cardiovascular disease or actually cardiac arrest, places where you really don't want to have almost a black box impacting efficacy. So yes, what we determine in terms of these rules of the microbiome and the impact on, just in the case of cancer, can be, equivalently applied to kind of unlocking how the microbiome impacts either the immune response or some type of chemistry that's involved with other types of therapeutics and used for a variety of diseases. Absolutely.
Unfortunately, that's all the time we have for today. If you have any further questions, please consider reaching out to the speakers directly. Their email addresses are shown on the screen. As a reminder, the webinar will be archived on the Scientist website. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the Scientist, I'd also like to thank our speakers, Christine von Raesfeld and Dr. Stephanie Culler, as well as our webinar sponsor, Sanguine. Thanks everyone, and goodbye.